You are warmly welcome to Virtual Science Education Center proudly present Biology Knowledge Hub program series. Today we hope to discuss episode number 7. And we hope to discuss about under our main topic is DNA and RNA. Our previous program we discussed about lipid and nucleic acids. If you want to still join our YouTube channel, please subscribe our YouTube channel and press bell icon. Also, I like to tell you, please tell your friends or other people to join and follow our YouTube channel and subscribe our YouTube channel. Okay, let's go to next slide. Okay, now you can see our discussion contain PowerPoint presentation home page. First, you can see our program series name is Biology Knowledge Hub. Second, you can see today we hope to discuss episode number. Third, you can see our main topic, and finally you can see today we hope to discuss subtopic under main topic. Okay, let's go to next slide. Okay guys, let's we see about today our lesson or episode learning outcomes. By the end of this episode, you will be able to describe below mentioned points. First one is definition and basic characteristics of DNA. Second one is building blocks of DNA and structure of DNA. Next, base pairing rule or Chargaff rule for DNA. Next, discovery of DNA. Next, location of DNA present in cells. Next, functions of DNA. Next, definition and basic characteristics of the RNA. Okay, let's go to next slide. Next, we hope to discuss today's base pairing rules for Chargaff rule for RNA. Next, building blocks and structure for RNA. Next, we hope to discuss types of RNA and their functions. Next, location of RNA present in cell. Next, we hope to discuss functions of RNA, and finally, we hope to discuss key differences between DNA and RNA. Okay, let's go to the next slide. Okay, now we start today's discussion. First, we discuss DNA or deoxyribonucleic acid. First, we see definitions and basic characteristics of DNA. DNA is one type of nucleic acid and it's a large biomolecule. Usually, it consists below elements in both nucleic acid. In the first, when to see the slide, you can see structure of DNA picture. DNA contains elements are first is carbon, next hydrogen, oxygen, nitrogen, and phosphorus. Small unit of DNA is deoxyribonucleotide, and that nucleotides are polymerized together using phosphodiester and hydrogen bonds to form these macromolecules. Okay, let's go to our next slide. DNA have two polynucleotide chains and double helix structure. Main function of DNA is gene information store and transfer to next generation in organism. And another function. Okay, let's we discuss about building blocks and the structure of DNA. When to consider that topic, smallest subunits of DNA is deoxyribonucleotide. Each nucleotide consists below main three subunits. That subunits are, first one is pentose sugar group. In DNA, pentose sugar group is deoxyribose. Next subunit is phosphate group, and another subunit group is nitrogen bases. When to consider DNA, there are four types of nitrogen bases. These are adenine, thymine, cytosine, and guanine. When to see the slide, you can see picture about deoxyribose, phosphate group, and nitrogen bases of DNA. Okay, let's go to next slide. Polynucleotide strands are formed using above mentioned units of DNA. Or that unit mean is deoxyribonucleotides. DNA have two polynucleotide chains and form double helix structure molecule. Okay, let's go to our next slide. Okay, let's discuss about base pairing rule or Chargaff rule for DNA. When two polynucleotide chains are bind to form double helix, there is a rule for pairing each nucleotides. Under this process, pairing happens between nitrogen bases in each nucleotide. When to consider DNA, adenine pairs with thymine, two hydrogen bonds, and cytosine pairs with guanine, three hydrogen bonds. Okay, and let's go to next. Okay, now we discuss about history of discovery of DNA. First, identified by Frederick Meister on 1869. Double helix structure was introduced by James Watson and Francis Crick on 1953. This model was introduced for describe what is a DNA molecule appearance. Rosalind Franklin's X-ray diffraction evidence were helped to create this molecule model structure. Okay, let's go to next slide. Okay, now we discuss about location of DNA present in cell, found mainly in the nucleus of eukaryotic cells as chromosomes can be seen, also present in mitochondria and chloroplast, small amount without cover protein. Okay, let's go to next slide. Okay, now we discuss about functions of DNA. Store genetic information of organisms. Next, transmit hereditary traits from one generation to the next generation. Next, provides instructions for protein synthesis. Finally, control cellular functions and metabolism. Okay, let's go to next slide. Okay, now we hope to discuss about another nucleic acid type is also known as RNA or ribonucleic acid. First, we discuss about definition and basic characteristics of RNA. RNA is another type of nucleic acid that plays a key role in protein synthesis. While this process RNA helps to transfer genetic information from relevant synthesis protein data contain DNA to the cell's protein making mechanism happen place in cell plasma. Now you can see in picture that slide, it is a picture of RNA molecule structure. When you see this picture, you can identify structure and appearance of RNA. RNA molecule consists below elements mainly carbon, hydrogen, oxygen, nitrogen and phosphorus. Okay, and let's go to next slide. The smallest unit of RNA is ribonucleotide. When RNA macromolecule is formed, these units are bind together using phosphodiester bonds and form single polynucleotide strand. When to consider RNA have single polynucleotide strand. Okay, let's go to next slide. Okay guys, let's we discuss about building blocks and structure of RNA. When to consider the smallest unit of RNA, it consists main three subunits. That subunits are phosphate, pentose, sugar group. When to consider RNA have ribose sugar. Second subunit is phosphate group. Third is nitrogen bases. In RNA have four types of nitrogen bases. These are adenine, uracil, cytosine, and guanine. Now you can see this slide. There are three pictures. First picture is ribose sugar molecule structure. Second is phosphate group structure, and finally have nitrogen bases structures. Okay, let's go to next slide. Okay, now we discuss about topic is base pairing rule or Chargaff rule for RNA. When to consider that topic, this base pairing rule is used for RNA mostly to RNA chain contain coding of protein synthesis process. Under this process, pairing is happen between bases each nucleotides. When to consider RNA uracil replace thymine and number form hydrogen bonds. When to form hydrogen bonds, two hydrogen bonds are formed between adenine and uracil, and three hydrogen bonds are formed between cytosine and guanine. Okay, let's go to next slide. Okay, let's we see main types of RNA and their functions. There are several RNA in our body. First one is mRNA or messenger RNA. First picture you can see structure of mRNA. Its function is carries genetic instruction from DNA to ribosome. Another RNA type is tRNA or trans-RNA. Second picture you can see structure of that. Its function is brings amino acid to ribosome during protein synthesis. Okay, let's go to next slide. Another RNA type is rRNA or ribosomal RNA. First picture you can see rRNA molecule structure. Its function is major component of ribosomes and helps in assembling proteins. Another RNA types are snRNA, miRNA, and siRNA. You can see second, third, and fourth picture to identify snRNA, miRNA, and siRNA. Function of these RNA are involved in gene regulation and RNA processing. When to consider RNA processing, it involves advanced types. Okay, let's go to next slide. Okay, and now we discuss about location of RNA present in cells. In this case, RNA found in both the nucleus and cytoplasm of eukaryotic cells and also present in ribosomes. Okay, let's go to next slide. Okay, now we discuss about functions of RNA. Functions are transfer genetic code from DNA to ribosomes. Next, directs protein synthesis, regulate gene expression. Some RNA molecules act as enzymes. Example, ribozymes. Another function is main genetic materials in some viruses. Example, HIV/AIDS. Okay, and let's go to next slide. Okay, now we discuss about main differences from DNA in RNA. First, we consider about pentose sugar groups. RNA have ribose sugar and DNA have deoxyribonucleic sugar. Next, we consider about nitrogen bases. Uracil in RNA replaces thymine in DNA. Next, we consider structure, usually single-stranded in RNA and double-stranded have in DNA. Next, we consider functions. RNA helps in protein synthesis, but DNA store and transmit genetic information. Okay, let's go to next slide. Okay, we come to end of today program. I think you can learn more knowledge and information about DNA and RNA. Okay, let's go to final slide. Okay guys, let's we go to finish today program. Thank you for watching our today program and stay tuned with us because I upload more videos about under this our program series, and I tell you please subscribe our channel and